Greetings, colleagues. My name is Kyle Boudreau, and I'm going to be presenting my paper of choice, which is Lactobacillus paracasei, KBL 382 administration attenuates atopic dermatitis by modulating immune response in gut microbiota. This was research conducted by Kim et al. 2020 and published in the journal Gut Microbes. So for background, why is this important? Atopic dermatitis, which I'm going to refer to from here on out as AD, has increased by roughly 1% per decade in children and adolescents. So this was published in a study, Langen et al. 2023. It's also worth noting that the prevalence of AD has also increased in adults over the period of time as well. And for me personally, I have a connection with AD. I actually suffer from AD. It's something I started developing in my late 30s. And here I have some of the worst moments. You can see some of the circles here highlighting especially some of the bumps, the rashes that one gets. So for me, it tends to be isolated to my forehead, fortunately, whereas other people can be affecting their hands or feet, other parts of their face, etc. How I chose this particular topic and this study, my best friend is a pharmacist, and he actually is the one who turned me on to Lactobacillus paracasei and AD because he had read a similar paper, I think published by a research team out of Japan, and had told me about it. So that, of course, piqued my interest. A little bit more, especially regarding the introduction here. So AD is an autoimmune inflammatory disease of the skin. Really at the heart of this are the helper T cells, for example, helper T cell 2. Without getting into all the specifics here, these are some of the things that we'll discuss that were addressed in this particular paper. So what pro-inflammatory cytokines, for example, are being further activated or released, the role of immunoglobulin E, and the different effects, particularly on the skin regarding scratching, itching, and water loss. It's also believed that dysbiosis is a major key player of dysbiosis of gut microbiota is a major key driver of AD, but this can be true at least regarding several diseases as they're being examined, obviously, in the current literature. So what is the role of dysbiosis in a variety of different, particularly diseases, and how does that affect those particular diseases? Lactobacillus or lacto-acid species in general have been shown to have benefits for improving especially health outcomes regarding these different types of diseases, especially inflammatory diseases. And especially modulators like the T regulatory cell or Treg, and especially the markers CD25 and FOXP3 are things that have been examined and are especially things that are examined in this study. And of course, regarding this particular study, they investigated the effects of lactobacillus strains that they isolated on AD-like symptoms in mice. And among the things they looked at, immune modulation with the Tregs, as well as the composition of the cecum microbiota, were things that were assessed. So for methodology, isolation and preparation, so what they looked at here, they took feces from Korean healthy adults, or healthy Korean adults, and they isolated two lactobacillus species, rhamnosus, which they refer to as KBL 365, and Lactobacillus paracasei, which they refer to as KBL 382. And I'm going to use those identifiers from here on out. The only thing they really mentioned regarding isolation preparation is that they cultured in Lactobacilli MRS agar supplemented with 0.5% L-cysteine hydrochloride at 37 degrees C, which should make sense. That's the typical average body temperature, and under anaerobic conditions, which is also typically the conditions of the gut itself, especially the large intestine. So here's a nice little image of a Lactobacilli MRS agar with some beautiful CFUs on it. And then the other thing they mentioned is they washed it with phosphate buffered solution, and PBS is something that's going to be mentioned from here on out as well, because it's used essentially as both a solution for administration as well as a placebo. And of course, sequencing analysis, I'll mention this in my discussions at the end, but I was very curious of why there was no mentioning of a sequence analysis to know that they specifically got these types, KBL 365 and 382. For methodology, experimental atopic dermatitis, so they used two known etiological agents that trigger AD or AD-like symptoms. The first one is Dermatophagoides farinae extract from this cute little creature here, which is a mite of the Dermatophagoides farinae type. And the other one is 2,4-dinitrochlorobenzene, DNCB. They also used 5-week-old male mice, specifically these NCNGA or Nga, probably pronounced mice. And from what I read, going all the way back to 1957, these particular inbred mice, these strains of laboratory mice, are the gold standard when it comes to doing this type of dermatitis analysis. For the groups, for the DFE-induced model, there were 4 groups with sample size ranging from 7 to 9. Group 1 was the PBS control, so they're just given PBS, no challenge with the DFE. Group 2 was challenge with DFE plus PBS. Group 3, challenge with DFE plus administration of KBL 365. And Group 4 was challenge with DFE plus the administration of KBL 382. All groups had hair of ear and dorsal region removed. And for groups 2, 3, and 4, DFE was administered with 150 microliters of 4% sodium dodecyl sulfate. Sodium dodecyl sulfate was used to essentially break down the barriers of the skin to make it easier for the DFE to get kind of into the deeper layers of the dermis, especially to trigger the AD-like symptoms. This was done 1 to 2 times per day over a course of 7 weeks. For groups 3 and 4, the associated lactobacillus strains, we're looking at roughly 1 billion CFUs that was suspended in 200 microliters of PBS, were given orally starting at week 3. For groups 1 and 2, PBS solution was given orally for 4 weeks. For the DNCB-induced model, it was similar to that of the DFE model. Here they only used 3 groups, and specifically they only looked at KBL 382, not KBL 365. Similarly, all groups had hair removed from ear and dorsal region. Here, forgive me, DFE was not administered. It was just strictly 200 microliters of 1% DNCB, which was dissolved in a vehicle of acetone and olive oil that was applied on the surface. And this challenge was done 3 times a day for 2 weeks. For group 3, I should say, the KBL 382 was suspended in 200 microliters of PBS and given orally starting at week 2, so similar to that of the DFE model. For the sake of time, because I've recorded myself several times, the last one was an hour long. We're definitely not going to talk about this. I will say that the results were very similar to that of the DFE-induced model. Methodology, so assessment of skin lesions and clinical dermatitis score. Mice were anesthetized, and then pictures were taken weekly of their skin from the beginning up to the end of the experiment. Severity of dermatitis was assessed, so they used 4 aspects or categories, if you will. So there was erythema, hemorrhage, scarring, dryness, edema, and excoriation erosion. And then each of these aspects were scored or ranked on a scale of 0 being no visible signs to 3 being most severe. And then they tallied all those together to get a total dermatitis score for that particular mouse. So that was the sum of all those aspects. So of course, if you scored 3 in all 4 aspects, you would have a total dermatitis score of 12. For TUL, which was the transepidermal water loss, as well as the moisture content of the stratum corneum, which is the outer surface of your skin. For TUL, they used the Tuometer TM300. And for the stratum corneum hydration, they used the Corneometer CM825. In both cases, the mice were kept at roughly room temperature, 21 to 22 degrees C, and at about average humidity, 50 to 55%. And these measurements were repeated 5 times. Scratching behavior, mice were placed in acrylic cases, and then they were allowed to stay there for an hour to get acclimated to being in these acrylic cases. And once the hour period was up, then a timer essentially was set for 10 minutes. So a 10-minute period is when they actually observed the scratching behavior. So after that hour, they watched for 10 minutes, and the 2 things that they measured were the number of scratches and the time that the mice spent scratching. For measurement of total immunoglobulin E, blood was obtained from the eye blood vessel. And here I had a question again. So I do have a background. I teach anatomy and physiology. So I know a little bit about especially immunoprivileged regions, which include the eye. So it's the only thing that could come to my mind because I couldn't find any good, clear answer, especially from the paper, why they chose specifically the eye blood vessel. But they did. So they drew blood from the eye, they centrifuged it, extract the serum from the top, and then ran the IgE and measured it through an ELISA kit. For measurement of cytokines and chemokines, these methods are a little bit above my head, so forgive me if I repeat this somewhat verbatim, but what I do want to point out is they essentially measured cytokines at the protein level, and then they also measured cytokines and chemokines at an expression, a level of messenger RNA. For the protein level, they used radioimmunoprecipitation assay buffer and protein inhibitors. And specifically what they're analyzing here were interferon gamma, IL-4, IL-5, IL-13, IL-10, and IL-17. So IL are interleukins, the specific class of cytokines. IL-10 is an anti-inflammatory cytokine, whereas the rest mentioned were all pro-inflammatory. The levels of cytokines were measured in the skin lysates using multiple magnetic Luminex kit. For expression, messenger RNA levels taken from the skin were measured using real-time PCR, and then that was run through specifically a Rotor Gene Q thermal cycler. For histology, what's notable here was the use of Congo red and toluidine blue. So Congo red was used because it has been shown to stain the granules and eosinophils, and toluidine blue was used to stain histamine and heparin and mast cells. So mast cells and eosinophils are especially two known cells involved with allergic inflammatory responses, and specifically they use further staining to identify those. Flow cytometric analysis. So again, I got to plead ignorance here, forgive me. So I can say that Tregs, I do know about them. So T regulatory cells are especially important in modulating the immune response. And here they took mesenteric lymph node samples. So the mesentery is the vascularized connective tissue between your intestines. And it also has dense population of lymph nodes. So it should make sense that that was a good source to take these numbers from, or these samples from. And specifically, they were looking at CD4 plus was mentioned, but really CD25 plus and FOXP3 plus were the protein markers for these Tregs. And they use a basic fixable viability stain, and then they use these more specific stains that stain the different types of cell differentiates like CD25 as well as FOXP3. For microbiota analysis, V3, V4 regions of 16S ribosomal RNA was collected from the cecum. They used barcoded primers, and then they ran these through a sequencer. In this case, they used I presume this is a MICSEQ platform, which is obviously an Illumina incorporated platform, a sequencer. Then they further did analysis using the KIME pipeline, which has been described obviously in lecture. And then the organizational taxonomic units, the OTUs, were set at 97% identity level, which were assessed using Green Genes database. For non-volatile acids and short-chain fatty acids, cecum was homogenized, centrifuged, and then the supernatant was removed or extracted. And then it was mixed first with a standard solvent, and then it was mixed with these more appropriate solvents. So for non-volatile acids, that was chloroform, and for short-chain fatty acids, that was ethyl ether. And then once they did that, they removed the organic layer, transferred that into a separate vial, and then that vial was placed into a gas chromatography to measure the different levels of these two acids, or these two groups of acids. For measurement of amino acids, cecum extraction was done similarly to what was previously described. Here, except the difference was that analysis was performed using ultra-performance liquid chromatography, which was aligned with analytical standard mixture and internal standard. And unfortunately, that's the extent of my knowledge about that. And the last thing I want to say is that statistical analysis, they used Mann-Whitney U-test, sometimes with some slight modifications, but ultimately throughout the entire experiments that they did, Mann-Whitney U-test was the one that they used every time. So for results here, we have an analysis overview of the DFE-induced model. So starting with the weekly analysis, again, we took body weight, dermatitis scores, and skin thickness. Then at 8 weeks, the poor mice were sacrificed. And then here, tissue collection, blood collection, and cecum collection were especially important, but they also did their TUL, their transepidermal water loss, and their corneometer, their moisture concentration of the stratum corneum measurements as well. Also, after those samples especially were collected, then they ran their histological examination, skin protein messenger RNA analysis of the cytokines, chemokines, flow cytometry, obviously, of the mesenteric lymph nodes for the Tregs, and then, of course, the cecum samples for microbiome and metabolite analyses. So for AD skin lesion assessment, if you look on the far left here, we have control, and you notice there's really no visible signs of AD. There's no redness, there's no lesions. Compare that to the far right here, which is KBL382. Some redness on the dorsal region, definitely some redness on the backs of the ears. Really no visible lesions that I can say, except maybe one or two spots. But where we especially see the skin lesions are with DFE PBS, as well as KBL365. And you can also see further darkening of the ears in both PBS and KBL365 as well. This graph just shows basically that dermatitis score. They were looking at the image assessment. Here they're actually looking at the score that they performed. Excuse me. KBL382 was the only one that showed significantly reduced numbers in terms of the dermatitis score. And worthy of note here is KBL365 as well as PBS were relatively even. Sorry, let me go back there. So KBL365 again, this was a lactobacillus strain that was administered. And here we can see really no significant difference. In fact, it was very close, very similar to the PBS, DFE PBS challenge group. For ear thickness, starting off, it's worth noting that there was relatively similar levels in terms of ear thickness between the three groups, 2, 3, and 4. But by the end of the analysis here, the only one that had significantly lower ear thickness, aside from control, obviously, was KBL382. For dorsal thickness, similarly, we see a similar trend, excuse me for being redundant there. We see very similar numbers in terms of PBS, KBL365, and 382 at the beginning of this, whereas towards the very end of this assessment, we can see that KBL382 had significantly reduced dorsal skin thickness compared to PBS and KBL365. Figures 2A through E are looking at a variety of measurements, including water loss, water concentration. So starting off, figure 2A is especially looking at the TUL measurements. So here, the only one that was significant in terms of lower evaporation or water loss through the skin, the transepidermal water loss, was KBL382. For corneometer, again, we want higher numbers here because this is the measurement of the amount of moisture in the skin of the stratum corneum. And the only one that was significant was KBL382. Numbers of scratches, so this was done again last in 10 minutes before the mice were sacrificed. And the only one with significant difference, although we can see KBL365 and 382 were lower than that at PBS. Really only KBL382 was significantly lower than that at PBS. Spleen body weight ratio, this one was interesting. They cite this particular paper, Han et al., 2012. And I went back and just kind of perused the paper. And indeed, what they found in that paper was that enlarged spleen was indicative of abnormal immune function. So that is why these particular researchers decided to also do that same assessment. So they used this previous study. And what they found basically was that KBL382 was the only one that had significantly lower spleen to body weight ratio compared to PBS and 365. The last one to mention here is immunoglobulin E and KBL382. Once again, if you're picking up on the trend, bravo. KBL382 was the only one that had significantly lowered levels of IgE. Again, elevated levels of IgE is indicative of an allergic or an inflammatory response. For histological assessment of mast cell and eosinophil infiltration, Here I'm just highlighting that these dark stains, you don't see the separate eosinophil versus the mast cells, but just highlighting, you can see these concentrations, these dark areas, which represent these different cell concentrations. And again, what's significant is KBL382 regarding lower levels of mast cells, and KBL382 regarding lower levels of eosinophils, these two cells that are especially involved with the inflammatory response. So higher numbers of mast cells and eosinophils, especially infiltration in the skin, is indicative of an allergic response. Here we see KBL382 has significantly lower numbers of these inflammatory cells compared to PBS and 365. Chemokine measurements. So what's really worth noting here is TSLP, because in a previous paper, TSLP has been linked with allergic diseases, including AD. So TSLP is thymic stromal lymphopoietin. Excuse me. And particularly keratinocytes, which are the cells of the skin, will release this as a way to activate and promote the proliferation of helper T cells, among other things, to promote the inflammatory response. So TARC and MDC, which are also chemokines, are also significant, but I just wanted to highlight that TSLP was especially something to highlight here, or something to point out. So KBL382, once again, significantly lower in regards to TSLP. For macrophage-derived chemokines, also was significantly lower compared to the other two. Here for TARC, which is thymus and activation regulating chemokine, KBL365 and 382 are both lower than PBS, but really only KBL382 was significantly lower. So this next analysis here, figure 5, has a lot going on. So what I'll highlight here is F, figure 5F, figure 5I, and 5J. These are the anti-inflammatory chemokines, whereas A through E, I should say, are the pro-inflammatory chemokines that were assessed at the protein level, and IL-2 and IL-31 were the pro-inflammatory cytokines that were assessed at the messenger RNA level. And what they found was that the pro-inflammatory cytokines regarding, again, KBL382, were significantly lower in all pro-inflammatory cytokines that were analyzed at the protein level. For messenger RNA expression of IL-2 and IL-31, once again, we see some similar trends between K365 and K382. Both are lower than PBS, but unfortunately, KBL382 is the only one that's significantly lower compared to PBS for both IL-2 and IL-31 messenger RNA expression. Measurements of anti-inflammatory cytokines. So IL-10 was the one that was measured at the protein level. And once again, KBL382 was, in this case, significantly higher. So we want higher levels of these anti-inflammatory, lower levels of the pro-inflammatory. So here, this is what one would hope for or expect when doing this kind of analysis. And we can see that among the three groups, PBS 365, KBL 382 is the only one that was significantly higher levels of IL-10. For relative expression and messenger RNA expression of tissue growth factor beta and FOXP3, again, KBL 382 was the only one that was significantly higher. Measurements of immune modulator from the mesenteric lymph nodes, the MLNs. So looking at particularly CD25 and FOXP3, what we can see is that both KBL365 and 382 were elevated compared to PBS, but really only KBL382 was once again more significant, or was significant, I should say. KBL365 elevation was not significant. For B, this is looking at particularly CD25 on the y-axis and FOXP3 on the x-axis, and what we see is a similar trend, even though KBL365 does show elevated expression numbers of these Tregs expressing CD25 and FOXP3. KBL382 is the only one that was significant. For average relative abundance of taxa at the genus level, here I'm going to first point you to comparing control to PBS. And what's notable here, we can see this category at the bottom. This is S24-7 unclassified taxa. And the other notable one here is this category, or this part of the bar, I should say, which is Bacteroides. And what they found was that with PBS, reduction in unclassified increased numbers in Bacteroides taxa was what occurred in the microbiota of group 2 mice compared to control. Contrastly, when you look at both KBL365 and KBL382, what they found was that unclassified as well as Bacteroides were actually closer to that of control. So figure 7b and figure 7c, we're looking at alpha and beta diversity, respectively. And what they found with the analysis of alpha diversity, 7b, was that really no noticeable differences, no significant differences in alpha diversity across all 4 groups. However, with figure 7c, with the analysis of beta diversity, what they did see was that there was actually significant beta diversity among all groups. Figure 7d, so this is significantly different taxa among different groups. So KBL365, for whatever reason, was excluded from this, but control PBS and KBL382 are emphasized here. And what the authors especially emphasized, what they felt was significant in this case, was the Akkermansia abundance, which I highlight here and which we'll talk about in the discussion. Comparison, figure 7e, comparison of relative abundances of significantly different taxa at the genus level. Again, what's notable here is Akkermansia. When we compare control to that of KBL382, PBS and KBL365 are relatively baseline. Well, some numbers, obviously some elevation, but not significant. Whereas if you look at KBL382 versus control, KBL had even higher numbers of Akkermansia abundance than that of control. So definitely significant in terms of the abundance of Akkermansia taxa in KBL382. Figure 8a is really just looking at the analysis of non-volatile acids and short-chain fatty acids. And what they noted here was that PBS versus KBL382, was that the environment essentially was conducive to cellular processes, genetic processes, information processes for PBS, whereas the environment was conducive to metabolic pathways and biosynthesis with the microbiome of within the cecum of control group 4, which is KBL382. For analysis of non-volatile acids and short-chain fatty acid, I should say, because really propionate was the only fatty acid that was looked at here. What's significant here is that we can see changes in PBS, but what's significant especially are the changes in KBL382 for both non-volatile acids and short-chain fatty acids. So elevated non-volatile acids and short-chain fatty acids in group 4, KBL382. Conversely, what we see are slightly elevated concentrations of amino acids, aspartic acid, serine, and methionine for PBS. For KBL382, even though 365 was lower in all 3 categories, again, it wasn't significant enough. So KBL382, significantly lower numbers of amino acids in all 3 groups. Excuse me. So for my discussion, really just kind of summarizing what we talked about, but I'm going to really focus again on Akkermansia to talk about that. First thing to highlight, obviously, is that AD-induced mice supplement with lactobacillus showed reduction in visible symptoms and associated behavior, the lesions, the thickness, the scratching. But the reality is even though there was some noticeable reduced scratching, especially with KBL365, it wasn't significant. Whereas with all those categories, dermatitis score, lesions, scratching, thickness, lactobacillus paracasei, which was KBL382, showed significant for all of those categories, significant reduction in those visible signs. Previous studies have actually looked at KBL365, which is Lactobacillus rhamnosus, and showed it to be beneficial for AD, interestingly enough. But in this particular study, it was really no significance in all the different tests that they ran. So without looking at those other studies, I'm kind of curious what those studies look like. I would look at them and see how they compare to that of this particular study. What did they do different that's comparable to this study, etc. So I'm kind of curious about that. KBL 382 showed significant amelioration of AD symptoms, which kind of ties to the first bullet that we mentioned. Notable were lower levels of pro-inflammatory molecules, cytokines, chemokines, higher levels of anti-inflammatory markers or cytokines. Alpha diversity was similar between groups, but beta diversity was significantly different. And again, what they emphasized here, what they also talked about in their discussion, which I'll also emphasize, that KBL 382 specifically showed an increase in the beneficial Akkermansia taxa. I read at least one paper that looked at Akkermansia and prevalence of obesity and type 2 diabetes, and particularly reduction in Akkermansia numbers, correlating to increases or an upward trend in those two diseases. And what they found was actually a correlation, a direct correlation between reduction in Akkermansia and increased prevalence of obesity and type 2 diabetes. So I think Akkermansia is definitely something that is obviously being studied, has been studied, and should continue to be studied, because it definitely seems like an important player in our, not only our gut microbiome and our gut health, but obviously in other factors, either directly correlated or otherwise to our overall health, including AD. Could be a possible connection there as well. So I think that's worth pointing out. Also worth pointing out is I did show that indeed there was increased activation of the immune modulation side of immunity, particularly the increased prevalence and activation of the Tregs and the mesenteric lymph nodes with KBL 382. So that was significant as well. So you have increase in beneficial microbiota, at least in this case Akkermansia, increase in immune modulators, reduced pro-inflammatory, increased anti-inflammatory markers, and obviously overall significant amelioration of the AD symptoms. So all that I think is very beneficial. Potential benefits of using lactobacillus strains as probiotics. So I think, again, as I mentioned before, when it comes to AD, you have some limited treatments, the use of antibiotics, the use of steroids. Those obviously are things you can't use long term. So you're kind of limited on things you can use for long-term treatment. So here, if this indeed is showing to be true, supplementing lactobacillus strains as probiotics might be the thing to do, obviously, with long-term treatment. For my discussion points here, first question is about sample size. I've never worked with mice. Maybe some of you have. I'm kind of curious what you thought about those numbers. Again, just they stood out as being low, but this could be par of course when it comes to mice studies. I don't know. The study had many tests, and indeed this is kind of the norm now in modern research. Do these big studies, multiple research agencies, universities, etc. involved, which I think is great, especially the collaborative side of all it. But on the other hand, sometimes I have to wonder, is this the best direction? I think sometimes small scale studies are equally as important, maybe sometimes better. So kind of just curious about what your overall opinion about these large scale studies that, like, for example, like this one that are conducted more and more nowadays. Why do you suppose they only used male mice in this study? So it could be a variety of reasons. Maybe that was the only thing they got their hands on. I don't know, but curious what your thoughts are. Akkermansia has been discussed, my apologies for that typo, in another presentation. So particularly, I believe it was Allison who did the presentation on personality traits that mentioned Akkermansia a few times. That caught my attention. So how significant do you think this genus is for our overall health in a variety of ways? Question 5, human trials, ? Can't sacrifice humans, can't remove their spleen, but you can do a lot of other comparable experimental tests similar to that was done here. And this is usually the progression, right? You start with maybe some in vitro studies, then you move up to laboratory animals, and then eventually move up to human clinical trials. So what do you think will happen there? How do you think human trials might compare to that versus this experimental design? Bonus question here, what do you think of their KBL strain isolation method? So again, that caught my attention, having some experience teaching some principles in microbiology labs where students had to isolate first using serial dilutions, different types of agar staining with different types of parameters, such as pH, salinity, etc. And then once they got their isolates down to what they thought was single CFUs, then we extracted DNA from those, analyzed those, and basically ran those in an NC blast. There was no mention of any of that in this particular study, so I'm kind of curious how they knew that they had these specific isolates. And that's it. All right. Well, thank you all for paying attention. If I went over, I do apologize. Good luck.